Okay, good afternoon, everyone. In the last class, we were discussing about transgenic animals. We already discussed about the direct method, and we were actually discussing about engineered embryonic stem cell method. One of the concepts from the last one is about how it is done in a lab completely, right? One is the molecular side of it, and the other thing is the animal side of it, or how are the organisms or the rats or mice used. So while we are focusing more on the molecular side of it, we should also understand a little bit about how it is done using the animals. So I'll quickly go through the direct method using how it is done in the lab. The same applies for engineered embryonic stem cells as well. So you need to keep in mind about it. So they superovulate a female, so that the female will produce multiple, eggs, and then they will allow mating with a normal male. Fertilization occurs, and then oocytes are generated. In the oocytes, as in the direct method, so the DNA construct is directly delivered into the male pronucleus. From the once those manipulated embryos are there, they are transferred into a pseudo pregnant recipient female. Okay, pseudo pregnant, meaning that this female is allowed to mate with a vasectomized male, so that the body of this female will feel much more physiologically and hormonally ready to retain the embryo. Okay, so the uh, 1 second, if you stay, please be online. I have to change my mobile. Okay. Um, so, am I audible? Right? I hope so. So, yeah, here is the preparation or the generation of pseudo pregnant recipient mice. Allow a normal female is allowed to mate with vasectomized male, so that it is physiologically and hormonally ready to retain the embryos, and then it would generate pups, and those pups will be analyzed or characterized for inheritance of the transgene. So in the last class, we were also discussing about positive selection and negative selection. If a presence of a gene is causing an advantage, is conferring an advantage, then it is positive selection. If the presence of a gene is conferring a disadvantage, then it is a negative selection. Here we use neomycin phosphotransferase that confers neomycin resistance. So that presence of that gene will allow the cell to grow in the presence of neomycin. Whereas in negative selection, we were discussing about thymidine kinase. The presence of the gene will kill the cell when it is grown in ganciclovir, a prodrug. So And we have established some things about when we are making the transgene, some rules. We want the uh, transgene W to be incorporated. We want it targeted. That means we want it only between X and Y, and it should not be anywhere else. Okay, and no other genes should be introduced during this process. So we'll try to solve some of the questions that we haven't addressed in the last class. So the goal is, I'll put, this is the goal, where this is gene X and gene Y, and assume this is gene W. So my goal is to put gene W between X and Y, and nowhere else, and no other gene should be introduced along during the process. So this is X. So we made a gene construct. The construct is something like this: X, then we had Y, because we want the homologous recombination would happen between X of the genome, something like this, as indicated here. And so this is W, and we need to have positive selection, because we should be able to select the cells that are transfected, the recombinants. So we will add, for positive selection, we will have NPT, neomycin phosphotransferase, the gene encoding for it. And we have, for negative selection, we have thymidine kinase derived from herpes simplex virus. So there will be three possibilities. Some cells will not get any. So we will then grow these under for positive selection, right? Here I will put as positive selection. So there will be three possibilities: cells that did not get anything, cells that got through homologous recombination, and cells that did not that got through non-homologous recombination. Okay, and I will just describe again what it means. This one is through homologous recombination. Homologous recombination would happen between the gene X of on the genome and on the construct. So these two will have a homologous recombination, and also Y and Y. So you'll have the integration of W and along with NPT for positive selection. Nothing else after Y is incorporated. Okay. Whereas in the through non-homologous means, you have X. Sorry, this is not X, right? Non-homologous. Sorry. That means it is integrating randomly in some part of the genome. So we would have, I will just say it as P, then you have X from here. All this construct that you are able to see, the whole construct of this will get incorporated here. From X till the thymidine kinase, everything gets incorporated. Let's just label them so that we'll have some clarity. And this is for, I'll put NPT, and this is Y, and this is thymidine kinase. And this one, assume it is Q. So we have three different cells: non-recombinants that did not get any of the cells, like here, then homologously recombined one, and non-homologously recombined ones. So we would have added neomycin in the growth condition, in the medium, growth medium. So homologously recombined one has neomycin phosphotransferase gene, and also non-homologously recombined one had a neomycin phosphotransferase gene. But the cell that did not get any of the DNA construct does not have. So this is minus for NPT. This is positive and positive. So the cells that did not receive anything would die out. What are selected? There will be two cells that will be selected: those that have homologously recombined and those that are non-homologously recombined. And then we would now perform negative selection. Negative selection is using a compound, a prodrug, gan-cyclovir, and there are also other names for it, but they act similarly. So ganciclovir is made into a toxic compound. It becomes toxic in the presence of thymidine kinase. Okay. So we are now adding, in the negative selection, we are adding ganciclovir. So we have two possibilities whether this will grow or not. Which one is thymidine kinase? Positive. This is TK minus, because as you can see in this case, the recombination happened between XX and YY. The thymidine kinase gene did not get integrated. Okay. Whereas the other one, the non-homologously recombined one, is TK positive. That's because you see that the whole DNA construct that we made is integrated randomly into some part of the genome. So you have all the genes, including thymidine kinase. Okay. So one of it is TK positive, that is non-homologously recombined one. The other one is TK minus. As we say, because the TK thymidine kinase would convert ganciclovir into a toxic compound, the cell is going to die out. So what we will be left with is the cells, these cells will grow and these would die out. That is how we would have achieved the target with this one. I'll come to in the few slides. I think this is until here we have learned in the last class, including this diagram also. Okay. So I think each of us should be aware of the components that are used, the selection procedure that is made, and the rationales of why and which one is going to get selected. So now again, as we have discussed earlier, I established that the goal is to have X, W, and Y. If you see what we got, this is what we got, isn't it? Right? I'm just depicting the same thing in this slide. Sorry, in this slide. So what I have got, X, W, NPT, and Y. I don't, as per the initial ones that I have established earlier, I shouldn't have this. This is an extra piece. I need to get rid of it. And how can we get rid of it? That is what will be the focus of today's class. And then that would be the end of this this particular lesson. Okay. So about removing, I also ask you to have a look at the site-specific recombination. Okay. In site-specific recombination, if you can recollect, you would have two different. You will have recombinase recognition sites. They are usually indicated like this. So these are kind of direct repeats. I will also draw one more for illustration purpose so that it will be easier for understanding. And these are inverted or opposite in the orientation. What are LOXP sites or recombinase recognition sites? You call them as recombinase recognition sites. So each of these actually has, I'm trying to blow it up so that I'll do it upwards so that it's easier to understand. If you read Watson or watched the animations, it would be much easier, but that's all right. So anytime you're doing recombinase recognition site, each of these recombination sites, okay, they're actually having three different parts. This is RRS, recombinase recognition site. This is also recombinase recognition site. And here you have crossover site. Okay. This one will also have similar, the other one also will have the same thing. In site-specific recombination, what one should always remember is what recombinases do. You should remember it is that recombinase enzymes, they would organize the recombinase recognition sites in this way: RRS and RRS. And the crossover site, so RRS here, recombinase recognition sites here and here, they have exact same sequence. What is, I mean, they both are repeats. In the crossover region, say for example, GACT is there. Okay, it's usually a little longer than that, but just assume that is there. Sorry, GTGA. Okay, GACT, I put it like this. So the bottom one would also have something very much similar. So enzymes, they can act on recombinase recognition sites when they recombination sites when they arrange this way: GACT, AGTC. Okay, I hope you see a polarity in this direction, and this one is in reading in the same direction. Just read the top strand, GACT, and read the other one in top strands, GACT. So it is like this. If I had a recombinase recognition site, let me move a little bit. I'll try to draw another one so that you will be able to appreciate what I'm implying here. So if I had the crossover region as okay, TCAG, and then AGTC. Okay, GACT. This is running in this direction, right? Of course, I'm reading it wrong. I should have read it 5' to 3'. What I mean to say is this is in the opposite orient. The sequence is opposite on orientation here. Recombination will only happen, site-specific recombination will only happen in this way. Okay, and the arrowhead indicates the polarity of these that I have drawn here. Just the small triangle that I put, that indicates the polarity. So in this case, the orientation is actually in the reverse direction to what I have drawn. So it should have been something, this is much more appropriate. This kind of combination is much more appropriate for this sequence. Okay, just assume this one. When we finish it, you will have a better understanding. So anytime for recombination should occur, you should have had these, you should be able to bend the DNA and should have it in the same orientation. Say for example, if I bend this DNA like this, and this triangle is in this orientation, and what would happen to this recombination site? It will also be placed in the same orientation, right? If I had genes A, B, C, D, okay, and I will say A, B, C, and D. Now, recombination would have happened between these two in this way. What recombinases would normally do is they would cut the DNA here and here, okay, and they would rejoin them in this way. If I stretch it out, I will try to draw a straight line. So I would have started with A, then I would have had a recombinase recognition site like I had before. Then this A, I will use a different color so that you will be able to follow. A is now following with, is continuous with C. So I have to draw it C here. That is continuous with B. And then it is continuous with the recombinase recognition site. It would have been something like this. And then that is continuous with D. So now if you compare this sequence with the resultant sequence, you will see that the B and C have got inverted, okay, due to recombination between the recombination site that are placed in inverted orientation. Okay. Now let's try to solve this one. First thing is, say I will try to put it, I will write genes also, P, Q, R, S. And I will try to invert them. And I'm drawing one of the recombination recognition site, the first one. And the second one, I am also trying to write here, P, Q, R, and S. Recombination cannot occur between these two. I mean, you shouldn't do it between these two because they are oppositely oriented. I hope you can, in this case, if you're able to see here and here, this is reading GACT and this is the in inverted position. Now, if the goal is anytime you want to make recombination, you want to solve problems on this, it's always good to put both of them in this kind of orientation. Okay. So if you are able to put it in this way, like this, then you are actually getting it right. So this is not happening. So what I should do is I should do something that nature actually does all the time. It is smart, right? Logic is, it's kind of always existing, or it's available to us. It's only about how we see it. Can we see it or not? Now, if I put, if you see that the arrowhead here that I'm showing is pointing towards R. So I should actually put this here. So compared to the previous one, I just folded like this, and then it was in inverted position. So if I could fold this whole thing more like this, then the arrowhead would be like this, isn't it? Once that is done, homologous recombination, site-specific recombination can be done here. What does recombinase do? Recombinase will cut these DNA molecules like this, and it will join, this one will join to this, and this one will be joined to this. Okay. Now let's stretch it out. What happens? We will check it. We'll see. I'll start with P. P, and then there must have been a recombinase recognition site, and that is continuous with S, right? Let's see what happens to Q. There is Q. Q is continuous with R, and R is continuous with Q. Only thing one has to add is that there is a recombinase recognition site here. What is the consequence of this particular event is deletion. In here, we had inversion. So how we place, where we place the recombination sites is will allow us to either insert, delete, or invert a DNA sequence. Now, why we have discussed all this is because until the previous slide, we have obtained engineered embryonic stem cells with an insert W. That's very good. It is homologously recombined. It is inserted only between X and Y and nowhere else. But there is a problem that we have NPT. We don't need that, or we shouldn't have it, because we don't know after it differentiates. If the cell differentiates, we don't know how it is going to behave. So we need to somehow get rid of NPT. And I hope from this slide you can figure out what should be done, right? We can use these recombinase recognition sites, and then we can try to, within the DNA construct itself, we should do it. So there are two possibilities in that way. And I want you to tell me which one would have been a good state to do. I will erase all these so that it'll be. Here is here is a thing that I had. So all the genome set, everything is same, but for so this is X, W, NPT, Y, and thymidine kinase. Now I'm talking about DNA construct. Okay, how should we design, and what kind, how should we implement a deletion system, right? There are two possibilities which one can try. One of it is, say, if I put this, if I put this between W and this, what would be the consequence? Can you quickly say, please? Anyone? If I put these are the black arrowheads are recombination sites. So we are actually discussing about these sites are called as LOXP. Okay, LOXP sites. It's a very common one that people use. CRE is the recombinase. The name of the recombinase, it's from Lambda, I think. CRE recombinase and LOXP sites. I think it is from one of the phages. So the sites are called as LOXP, and the recombinase name is CRE recombinase. CRE LOXP system, they keep saying. You will find in any in majority of the genome editing cases, you would find this thing, CRE LOXP system. So if in this situation 1, what would happen if I have introduced these LOXP sites in this particular orientation? You can try to answer, and then we'll see how far we get. Sorry. Okay, let's see what would happen in this case in 1. As I said before, these two are in opposite orientation. In opposite orientation, until I erased, I think you noticed that it would have, if recombination had happened, CRE recombinase was expressed, then it will just invert, and it will make a setting something like X N W Y T K. If we just added recombinase to the gene construct that we are making. Okay, if it is in inverted orientation. If we put in something like this, if we have put it in this way, what would happen? Then it would be deleted. One second. Yeah. Okay. Anyway, if I put, as in the situation 2, if I put it like that, what would happen? Then it would form X, Y, TK. W and N would be deleted. Our goal is to just delete NTP, right? So there are two things that we need to remember how we introduce the LOXP sites. Where are you putting, and in which orientation you are putting? It should be put in the same orientation like this, right? But if we do the placement, look at the placement. If you put it in here between W and N, then we are getting the deletion of W. We only want deletion of NPT. So what we must actually do is that this should be placed somewhere here. Okay, then what are we getting? This would be the ideal, our complete construct. I should, so please remember that this is the second one is the correct one. I will review quickly the genetic elements. So try to keep up and see if you have any questions. So X, this is the description for gene construct. Okay. And we will also get to a situation where we will try to discuss, we will assume that we have already introduced, and then what would happen. Okay, these are the recombinants we got in the last slide. So X is for homologous recombination because we want a targeted gene insertion. W is the transgene that we intend to integrate between X and Y. That is the reason why we have introduced X and Y. I have used neomycin phosphotransferase gene here because I want to have positive selection. This is for positive selection. I should be able to select those that are recombinant from those that are non-recombinant. And then I have used thymidine kinase because I want it for negative selection, because I want to eliminate all those that would have integrated through non-homologous means. Then I have also introduced 2 recombination sites in the same orientation because I want to get rid of this neomycin gene at a later stage. If in an exam, I might, or anybody might, I typically would have asked, I would have scrambled things like anything, in the sense that X, W, Y, N, and thymidine kinase. If I asked you to find out what are all the mistakes in this, okay? Yeah, let's just put up things. What are all the mistakes in this gene construct? First thing is, yeah, until these 3, this is fine, okay? But then everything else is wrong. I mean, thymidine kinase placed outside is all outside the genes of homology is also good. That is also correct. What is wrong is placing neomycin phosphotransferase gene outside this boundary, the X and Y boundary, is wrong. Why is that? If integration happened, you will get X, W, and Y, but you will not get neomycin phosphotransferase. And in the selection, in the positive selection, if you have used neomycin, all those that have recombined homologously would die, will not be selected, because neomycin never got integrated. But you will get selection if any cell is growing, that means that must have integrated non-homologously. Somewhere random. We are losing the specificity or the target between the X and Y. And we are also, I mean, that is itself is gone case. Second thing, that is the reason why positive selection should always be placed within the boundaries of the homology. It is the targeted region, X and Y. The second thing is the placement of the recombination sites. They are in proper orientation. That is good for deletion in that sense. But then they are actually, once they are integrated, they would actually, it might not happen at all. The integration site, second recombination site, is outside the region of homology. And important one more is we are using LOXP site or restriction LOXP system, Cre-LOXP system, to get rid of neomycin. And therefore, this placement of this recombination site between X and Y is wrong. Okay? In a typical exam, a question would come where you are supposed to evaluate a gene construct, and then you should be able to do it with proper reasoning. Okay? Good. Any questions you can ask if there are? Otherwise, I will complete this lesson. Okay, I hope you understood that. So what would happen here is, you got these cells. These cells have this setup, and now our objective is to get rid of NPT. So the cells will have transiently, so from an episome or so, you would express Cre recombinase. Okay, assume this whole genome setup is there. I'm only drawing NPT here, which is with the recombination sites on the its same orientation. So Cre recombinase is expressed. The enzyme, the transposase would bind here and here. The DNA is looped, and then these strands are exchanged, because of which neomycin, this NPT, comes up. It gets deleted, and ultimately, what you would have is something. Let me check. So ultimately, what you will have is you will have the deletion of this X, right? This part NPT will be gone because you have expressed Cre recombinase. What you will be left with is the one recombination site will be there. It's not a gene. There might be some effects, but it's negligible. That is the least bargain one can have. There is a footprint of one of the recombination sites left there. But otherwise, you have X, you have Y, and there is W. We have fulfilled the objective of inserting W between X and Y, and it is not integrated anywhere else. And second, the last one condition is that we did not insert any other gene. Okay, LOXP is not a gene. It's just a small sequence of some say 10, 15, or 20 bases at the most. But that's a compromise we can go with. Okay. So in this last part, we should understand. So we have taken the inner cell mass. We got the embryonic stem cells. From the embryonic stem cells, we have transfection. We have done the transfection. Then we have been to positive selection. Then we have negative selection. Then we have Cre-LOXP system. Sorry, LOXP system to get rid of the positive selection marker. And now we have engineered embryonic stem cells like we want. Now what we do is we take these cells and we put them into a blastocyst. So the recipient blastocyst now has two types of ICM. One is the ICM from itself and the ICM from engineered embryonic stem cell, right? And it is implanted into a recipient mouse, and this mouse would have been prepared like pseudo pregnant, like I have described before. And then it would give rise to several pubs. In some of them, integration could be more. I mean, we will do this blastocyst manipulation. Multiple blastocysts are done and put into one pseudo pregnant mouse. And then we will have, this is not yet a transgenic founder. We will have actually a chimera. Okay, we will describe what is a chimera and what is not in the following. Here, the same thing basically. We have done the manipulation, genetic manipulation. We have introduced these cells into a blastocyst. We introduced into a mother, foster mother, and now we get a chimera. What is a chimera? I think I described in the last class. I'll quickly go through it, at least in the context of what we are learning now. We have one blastocyst, assume that is having green ICM. We are now introducing the engineered embryonic stem cells into this, okay? So we have some cells which are ICM given like this. The whole ICM is going to give rise to one animal. Okay, so the total of it is all this whole mass is going to give rise to the mouse. Some parts of it will be derived from the green ICM like this, and some parts of it would be derived from the red one, red ICM. If you remember the definition of what is transgenic animal, we discussed that the transgene should be present in all of the cells. Okay, if you see this mouse, which is a chimera, because some body parts are made up of red ones and the others are made up of the green type of green ICM, just for understanding sake. So not all the cells, only the red ones actually have the gene of interest that we are looking at. So chimera is not yet a transgenic. So we should now allow this to mate with a mouse that is completely green, normal mouse. Okay. And when they have these, if there are, if this mouse has, or every organism has somatic cells, somatic part and germline, germplasm and somatoplasm. There are two possibilities in this chimera, or three possibilities. It is possible that in terms of germline, all the germline could have been made by the green one. It is also possible that all the germline is made by just red cells. It is also possible that it is made up of green. Some parts of the germplasm are made up of green and some parts are made up of red. That is also possible. So now these are three possibilities, and we should be able to derive from the answers how what is there. So I will give three conditions, and from those conditions probably you can figure it out. Assume that when the mating between these two organisms has happened, the chimera and a wild type mouse, green one completely, and if all the mice were red in color, what would that mean? Say they have two babies, pups, and they are all made up of, they are all red in color. What does that mean? If some of them are made up of green and some of them are made up of red in color, assume that the transgene is the dominant trait, okay? Just for simplicity of understanding. All are green. So I'm talking about three independent events, okay? If you have this, you may have this situation, or this situation. In situation one, all the offspring are red in color. In situation two, some of them are red in color, some of them are green in color. In situation three, all of them are green in color. Then which one is the transgenic here? Which are all the cells that are transgenic? The animals. We introduced red transgenic or engineered embryonic stem cells into the blastocyst. So we are looking for a body or an organism that is completely red. This is a transgenic, and this one is a transgenic, and so is this one. Okay, all the green ones are non-transgenic ones. So what you can, if you had all the offsprings are red in color, that means in this chimera, all of germplasm is derived from the implanted engineered embryonic stem cells. If it gives some green colored ones and some red colored ones, then it would mean that in this chimera, the germplasm is made, some part of germplasm is made from green cells and some from the implanted engineered embryonic stem cells. If it is all derived from, all the pups are actually green in color, then the whole experiment actually failed. Again, people have to go back to the designing of the gene construct, look into it, and then evaluate and do some stuff. Okay. So that means in this chimera, if this is situation 3, it indicates that the germplasm in the chimera, all of the germplasm in the chimera is derived from the green ICM. Okay. I hope you understood the questions. if you have any questions here, I would like to take. We have 10 minutes, please, if you have any questions. But that concludes the transgenic animals. Applications and other things you have to read. I'm more interested in the process and the technical aspects of it. The other applications in disease models and many other things, you can read that up in textbook. Any questions? Okay, so I'll conclude and then I will probably see what can be done. So in transgenic methods, we have seen in animals, we only focused on animals. In plants, there is again interesting one with gene gun. You can also do it with in animals too. There is also agrobacterium mediated gene transfer. That is an exciting one in plants. You have to, agrobacterium has a very interesting, with the TI plasmid, it's an exciting field. But that's not in the syllabus, so we won't discuss that. So we have discussed two methods. One of it is the direct method, where we directly inject the gene of interest into male pronucleus. So hoping that it will integrate and in only one copy, but that's not usually the case. It is very simple. It is very efficient. Okay. The problem, however, is that the integration is random. It's not targeted into one particular site. And the second problem is there are numerous or multiple copies of it integrated, not just one copy. And that gives, it gives a lot of uncertainty about what to do, and we have to characterize. And after characterizing, we might think that it actually, the integration may have caused a defect in one or more of the genes, because of which it might become unuseful to us or useless. There is also the second, we also have discussed about retroviral methods and so on, where it is very efficient because we are choosing the infection model of the virus itself. Single copy insertions are possible, and it's very not so much of technical equipment is required. Say, compare it with this direct microinjection. You need to have a manipulator, micro manipulator, and the suction pipette and all this stuff. But with viral infection method, it's much more easy. The problems, however, are these. The mortality rate is very high, because partly also because of random integration. And infection does kill the cells. That is another problem. And there is also packaging limit based on the capsid and the head particle of the virus that it would form. Similar to that, we have learned in lambda phage, for example. We had a limit. The head space can only accommodate as much. Similarly, here also they have some packaging limit. If we want to use for clinical purposes, and if we have employed viral particles, that's going to be a big mess, in the sense that it is prone to risks, because although we have removed majority of the viral genes, the sequences may allow further recombination. Some issue may happen, and that's always a scare that is present with using viral vectors for clinical purposes. So the next one is engineered embryonic stem cells. We can target the genes. We can ensure that it is single copy insertions. Long transgenes are also possible. The problem is that there are a lot of technical difficulties. You should have a cell culture growth system, and you need to maintain the embryonic stem cells for a long periods, and that's technically much more difficult and time consuming. And we should have there are some, whether we have this mouse cell line for this or not, and some other technical issues are also related. But This is the best method for clinical purposes. In clinical purposes, meaning that we are going to employ these for humans, say that is the idea or goal of it, then we want accuracy is very important. If we make a mistake, instead of treating the patient, we might actually cause complication or even death. And that is always something the clinical trials and everything will be focused on. And for that reason, engineered embryonic stem cell is a very, very good one. But now, as we will study in the later part, we will read about CRISPR-Cas9 system, and that's an exciting one. Before going into that, we will have to learn about the paper that I already sent you, that is transgene-free generation of induction of pluripotency. So please read that. I will try to finish that soon as well. And then I want to get into CRISPR, and it'll be because I want to discuss in basic science perspective also, and then applications. Okay. So quickly, very small note, and I was describing about spatial and temporal, right? When you put a gene W, you want it to be expressed, and therefore you should have had a promoter. And the choice of the promoter is usually tied to your objective, right? Your objective could be like, I want expression of W in neurons, or it could be I want expression of W in epiblast, a growth condition, differentiation during embryogenesis. So you're choosing where the expression should be, or you might choose when the expression should be there. And for that reason, what will become very essential is the regulatory sequences that you have put. What kind of promoter did you put? So in this particular figure, they made transgenic mice, and they want the gene to be, this particular gene to be expressed. Say, for example, they have used a marker gene for, I think it would be like Z or so, and they want it to be expressed in some place, and then you should have used the promoter that would have been active in that particular tissue. That means you should have used tissue-specific promoter. What I mean by that is that promoter would be active only in that specific tissue and nowhere else. Here, they have used a gene. Yeah, it is beta-galactosidase. That's right. And then they would have put, because in the result that is being shown here, they have used laxate, and they have used the promoter of, it would be, okay, neurogenin 1. Okay. So this promoter is active only in the neurons. So you can see that the brain and you can track the development of nervous system, where the nerve cells are there, and so on. This is just one example of many examples that are there. The people use GFP a lot. You can use GFP and use the promoter, which will allow proper expression of the gene where you want, when you want. Okay. Any questions? You must have something here. If you have any questions, please let me know. And I hope you are all applying for GATE. Please be proactive, take chances. Nothing wrong if you didn't qualify. Apply any exam that you are eligible for. That's the strategy that works. That is a decision where you take pro work, for work, not to, you have no reason why you shouldn't write GATE actually. Even if you're choosing to go abroad, or you're trying to write exam, go for job, On an application, if you had something like get qualified, it's a good indication of your standards, of your degree, and your worthiness. Even if not, first thing is to have the guts to apply and say that I want it. The desire should be there to start with. Once the desire, you will have the guts to apply for it. Once you have the guts to apply, if you have applied for it, you will study a little bit. Even if you don't qualify, that little bit of study is going to become useful to you. In the past, there were several people who have written just like that and got qualified, and they have they put it on their CV. Okay, they didn't aim for it, they didn't prepare also. Sometimes they went and wrote and they qualified. Your education is good enough. All you need is to know what that exam is, go and attempt it, and then I'm sure you will qualify. Okay, I think we should be much more proactive in that sense. I forgot about GATE exam, so on. That's why I'm telling it now. Somebody told me this, and then I recollected that it was. Anyway, the date is extended a little bit. Try to apply and take more chances. Okay. Thank you very much.